Classification of carbohydrates. Carbohydrates can be classified as monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharide represents the monomeric unit. They can be classified into two subclasses. Based on carbon length, they can be classified as triose, tetrose, pentose, and hexose. Based on functional group, they can be classified as aldose and hexose. Examples for monosaccharides include the triose contain 3 carbon atoms, and the function group aldose contain aldotriose is glyceraldehyde, and the ketotriose is dihydroxyacetone phosphate. Tetrose has 4 carbon units, and tetrose is erythrose, and ketotetrose is erythrulose. Pentose have 5 carbon atoms. Aldopentose is ribose, and ketopentose is ribulose. Hexose represents 6 carbon atoms. Aldohexose is glucose, and ketohexose is fructose. Disaccharides are made up of 2 monomeric units linked by glycosidic linkage, which can be classified into 2 types: reducing and non-reducing. Reducing include lactose made up of glucose and galactose, present as component of milk sugar, and maltose made up of glucose and glucose. Non-reducing sugar are glucose and trehalose. Glucose is made up of sucrose and trehalose. Sucrose is made up of glucose and fructose. Now these glucose and fructose are linked by glycosidic linkage, which involves a functional group, hence sucrose is non-reducing. Next, trehalose is made up of 2 molecules of glucose. Oligosaccharides oligosaccharides contain 2 to 10 monomeric units linked by glycosidic linkage. Now these oligosaccharides act as cell surface receptors for entry of biomolecules like hormones. They also act as cell surface receptors for entry of pathogens like bacteria, virus, fungus, protozoa. Apart from this, oligosaccharides also act as blood group antigen present on the surface of erythrocytes. Next are the polysaccharides made up of more than 10 monomeric units. Polysaccharides can be classified into homopolysaccharides and heteropolysaccharides. Homopolysaccharides are made up of same monomeric units, whereas heteropolysaccharides are made up of different monomeric units linked by glycosidic linkage. Examples of homopolysaccharides include starch made up of amylose and amylopectin, which is the storage form of carbohydrate in plants. Glycogen is made up of glucose linked by 1,4 linkages and branch at 1,6 linkage, and the glycogen is the storage form of carbohydrate in animals. Next is cellulose made up of glucose linked by beta glycosidic linkage, and cellulose is the component of dietary fiber. Inulin is made up of fructose in beta linkages, and inulin is mainly used to estimate glomerular filtration rate. Next is dextran made up of glucose with alpha 1,4, 1,3 and 1,6 linkages, and these dextrans are used as plasma volume expanders, and they are used in the treatment of shock. Coming to heteropolysaccharides, which contain different monomeric unit. The examples include the mucopolysaccharides, also called as glycosaminoglycans, which contain repeating unit of disaccharides. One amino sugar will be there, and the other one is uronic acid. The examples include hyaluronic acid. Composition is glucuronic acid and glucosamine, mainly present in vitreous humor and synovial fluid, and acts as lubricant. Chondroitin sulfate, glucuronic acid and galactosamine, present in cartilage, tendon and ligaments. Heparin made up of ioduronic acid and glucosamine. It acts as anticoagulant. Dermatan sulfate, composition is ioduronic acid and galactosamine, present in skin, heart valves and blood vessels. Keratan sulfate lacks uronic acid but mainly contains glucosamine and galactose. It is responsible for maintaining the transparency of the cornea. It is one of the component. So this was about classification of carbohydrate.